Lab 16, Cellular Respiration. Introduction. The process of respiration. Cellular respiration is the process that converts glucose into ATP, a form of energy that cells can use. The chemical reaction for the formation of ATP is adenosine diphosphate plus phosphate plus energy equals adenosine triphosphate. A chemical formula for the release of ATP energy can be written as adenosine triphosphate equals adenosine diphosphate plus energy plus phosphate. Energy is stored in the bonds between phosphates, with the greatest amount of energy in the bond between the second and third phosphate groups. When the third phosphate is cut loose, ATP becomes ADP and the stored energy is released. The input of additional energy plus a phosphate group recharges ADP into ATP. Glycolysis. Glycolysis is a universal process. 9 reactions, each controlled by a different enzyme, make up the process. All organisms have glycolysis occurring in their cytoplasm. Glycolysis begins when ATP is converted into ADP, inputting energy into the reaction. The energy is used to convert glucose into pyruvic acid. The end of the glycolysis process yields 2 pyruvic acid molecules and 4 ATP, for a net gain of 2 ATP per glucose molecule. Anaerobic and aerobic pathways. Pyruvic acid can then be routed into one of 3 pathways: lactic acid fermentation, alcohol fermentation, both are anaerobic, or cellular aerobic respiration. These biochemical pathways, with their many reactions catalyzed by enzymes all under genetic control, are extremely complex. Alcohol fermentation is the formation of alcohol from sugar. Yeast, when under anaerobic conditions, converts glucose to pyruvic acid through the glycolysis pathways. Then go one step further, converting pyruvic acid into ethyl alcohol. Many organisms will also ferment pyruvic acid into other chemicals, such as lactic acid. Humans ferment lactic acid in muscles where oxygen becomes depleted, resulting in localized anaerobic conditions. This lactic acid causes the muscle stiffness couch potatoes feel after beginning exercise programs. The stiffness goes away after a few days, since the cessation of strenuous activity allows aerobic conditions to return to the muscle, and the lactic acid can be converted into ATP via the normal aerobic respiration pathways. Alcohol and lactic acid fermentation dispose of the pyruvic acid but produce no energy. When oxygen is present, aerobic conditions, most organisms will undergo 2 more steps: Krebs cycle and electron transport, to produce more ATP. This process occurs in the mitochondria, a structure found in the cell, and is known as aerobic respiration. This lab will explore the rate at which cellular respiration occurs by measuring the amount of carbon dioxide produced. Materials needed: dry yeast, sugar solution, a glass jar, 250 milliliter glass beaker, stopper with 1 milliliter plastic pipette, and a stopwatch. The procedure: 1. Obtain a half a gram of dry yeast and record this data on table 1. 2. Fill the 250 milliliter beaker with the warm 1% sugar solution from the supply table. 3. Place all of the dry yeast in the jar. 4. Before stoppering, fill the jar with the 1% sugar solution. Add sufficient solution so that when the rubber stopper is inserted into the jar, there is no air trapped under the stopper. 5. Adjust the rubber stopper so that the water level is at the 0 line. 6. Record this beginning level on the table. 7. Record the fraction of a milliliter The fluid on the tube moves up every 3 minutes for 15 minutes. 8. Calculate the production rate of carbon dioxide over the 15-minute period. Record this information on Table 3.